Okay, hi everybody. My name's Paige. I'm a master's student here at the University of Waikato, and I will be sharing some of the results from the first chapter of my master's research, which is on how genomic data is missing for many highly invasive species, which is restricting our preparedness for escalating incursion rates. So I'm sure that this is not news to any of you, that invasions of all taxonomic groups have risen steadily over the past few decades and will likely continue to do so as things like climate change, globalization, and land use change facilitate the invasion process from introduction right through to establishment and spread. And so, of course, concerns are growing about how these will impact future things in the environment, biodiversity, human livelihoods, and local and global economies. So far, a lot of invasive species management relies on reactive responses, such as eradication, and this is really our only option a lot of the time because we don't really know the full extent of a species' invasiveness until they've pretty much spread everywhere and ruined everything. Predator Free 2050 has done some really great work at trying to eradicate some key mammalian pests in New Zealand, but it's not exactly a practical way to deal with every single invasion in the future. And so being able to predict and prepare for invasions will almost certainly lead to better overall outcomes. But how can we work towards this kind of approach? Well, it is really difficult. How do you know if a species will become invasive? Invasions are so complex and depend on so many different things, of course, physiology, ecology, but also things like adaptive potential and plasticity and things like that, and how these all interact with humans and the environment, and not to mention how they will evolve over time and space. Also, some introduced species have lag periods, which may last for many, many years, which complicates the process even further. And there's just a whole lot of unknowns. And so this has been a really big question in invasion biology and limits our ability to be prepared for invasions and act proactively. In the past, we have used genetic markers such as mitochondrial DNA or microsatellites to make inferences of populations. This is also known as population genetics, which I'm sure you also all know about. And this has given us a broad understanding of the general effects of things like bottlenecks and genetic drift on invasion success. Population genetics is a really valuable tool, and it has been for many, many years. And it's also paved the way for population genomics. Moving to a genome-wide or genomic scale, as opposed to just single or few sets of loci, can improve analytical accuracy in some scenarios and also allows us to address some research questions in much higher resolution. Briefly, some applications include looking at the population structure of invasive species at small and large scales and to see how populations are connected or related to one another. Looking at the genetic effects of invading populations, such as founder effects and how this shapes their invasive potential. And something I personally find really cool is, and probably the thing that sets population genomics apart from population genetics the most, is the ability to characterize specific genes and the roles they have in invasion success. And I think Ellie's going to talk about this more tomorrow. Yeah, so stay tuned for that talk. It's really cool. Okay, and so with this in mind, population genomics has heaps of potential, right? But to what extent is it actually being used to study invasive species? And this is what we aimed to find out. So this is the International Union for the Conservation of Nature 100 of the world's worst invasive alien species list. I will just call it the invasives list because the name is really long and I don't want to get tongue-tied. This list was curated in 2014 and aims to increase awareness of invasive species and their impact on the environment. Species are chosen based on their impact to biodiversity and human activities, and also their illustration of important issues of biological invasion. Only one species per genus are included to ensure that there is a diverse representation of taxonomic groups. So you can see that this ranges from microorganisms right through to vertebrates and mammals and stuff like that. So for every species on the list, we did a systematic literature search using these criteria. Does the species have population genetic data? Does the species have population genomic data? Does the species have a reference genome? And if yes, was this in an invasive context? So first we looked at the general uptake of population genetics and population genomics in the literature and found that generally speaking, publications that use genetic markers to study populations still dominates the literature and has done for the past 20 years. You can see that around about in 2009, population genomics sort of starts to take off, and this correlates really nicely with the decreasing cost in sequencing, which was about $1 American back in, per raw megabase back in 2009, and is now about 0.006 American dollars. It's great to see progress in this space, but there is still probably financial and computational burdens that limits the amount of genomic studies we're able to do. In regard to the worst invasives list, we found that the majority of these species, 74% in fact, had been studied using population genetic data in an invasive context. However, the invasive context was limited. Most studies focused on the history or the route of invasion or the demography of the colonizing population, and very few studies have looked at the evolution of invasiveness. Of the 18 that didn't have population genetic data, 13 were plants, 4 were invertebrates, and 1 was a macrofungi. In contrast, we found that only 24% of species on the worst invasives list have population genomic data in an invasive context. Of these 24, you can see that the evolution of invasiveness is a bit more balanced with the other contexts, which is good because there's growing evidence that underlying genomic changes have big roles in invasive potential. But there's clearly more work that also needs to be done in this space. Next, we looked at how many species had publicly available reference genomes. In 2019, 27 species on the worst invasives list had a publicly available reference genome. And then in 2022, we found that this number increased to 45, which means that 18 were sequenced in the last 3 years and indicates a growing investment in genomics. And as I was creating this presentation, I also noticed that 2 more species have been sequenced or uploaded to NCBI, which is great. Lastly, we looked at taxonomic trends for reference genomes. So the outer ring represents the proportion of species on the worst invasives list, and the inner ring represents the proportion of has a reference genome versus doesn't have a reference genome. So for the groups that only had a few species represented on the list, most of these had a reference genome with the exception of a few birds, fish, and the brown tree snake. Mammals made up 14% of species on the list. Just 3 lacked reference genomes back when I did this analysis in May, and this number is just 1 with the small Indian mongoose who's still yet to be sequenced. Invertebrates made up 26% of the worst invasives list, and half of these lacked a reference genome. And finally, plants made up the majority of the worst invasives list, which was 37%, and an overwhelming majority lacked a reference genome. And these results don't necessarily reflect the relative impact of each invasive group. I mean, 37 plants on the worst invasives list, they probably wouldn't have made it on there unless they were pretty bad invaders. But it instead probably reflects like taxon-specific idiosyncrasies, such as the fact that plants have really long, complex genomes with lots of repetitive content, lots of polyploidy and stuff like that. But thankfully, long-read sequencing is helping to bridge this gap. And so hopefully in a few more years, we'll see a lot more plants being sequenced, which will be really great. And so what's next? Well, hopefully we will see more, an increase in high quality, annotated and publicly available reference genomes. There are some projects such as the 10kp, 5k and the Earth Biogenome Project, which are starting to do stuff like this, which is awesome, but it would be cool to see more targeted invasive species initiatives. You don't necessarily need a reference genome to study invasive species. A lot of really good studies have been done without the use of a reference genome, but it really is a game changer for being able to characterize genes involved in invasion success, which so few studies have targeted so far. This one here did a genome scan to identify outlier SNPs under selection, and they had a really high quality reference genome, which meant that they were able to identify particular genes that were involved in invasion routes for this fly. And we also want to see population genomic data being used for more targeted invasive species management. For example, this study analysed patterns of connectivity for 2 rat species that kept re-invading an island with vulnerable seabird species and managed to identify different eradication units that could then be prioritised for management. All in all, it is clear that there is a growing investment in genomics, which is great, but there's still heaps of work to be done. It's a really cool field to be researching in, and I'm super excited to see where it goes from here. Special thanks to the Invasive group, especially to Ange, my supervisor, and yeah, that's it. Thank you.
Thanks for that, Paige. Has anybody got any questions? I have one. Start. So you're saying that 55% now of the invasive species on the top 100 list have a reference genome. Do we know whether or not these reference genomes are from native or expanded ranges? Or are they from like the invasive population or the home population?
It's just all over. So
yes,
either or, yeah. So yeah, that's a good point. Yeah.
Yeah, I think it would be quite cool if we knew, and then we could have a comparative.
Yeah, that's very true.
Yeah.
Thank you. That was really fascinating. I have a couple questions. One is, I'm wondering if you distinguished between like a really good genome, like chromosome scale, or just anything that somebody said was a genome assembly. Yeah. And I'm also wondering if you noticed any patterns in terms of, the genome complexity. So I know like, plants have often really polyploid genomes. And so I don't know if you looked specifically at this or if you anecdotally noticed patterns in, easier genomes like birds being much more sequenced than genomes that might be a lot more complicated.
Yeah, we did take notes of what the amount that it had been sequenced. A lot of them were just scaffolds, not a lot of them were at the chromosome level. So yeah, that's definitely another thing that we noticed.
Sure. Yeah, I know that's something I've run into trying to do adaptive work is that so it's not even if they're not really well assembled genomes, they're quite not terribly useful.
Totally, yeah, that's definitely a limitation as well, yeah.